Hello everyone, welcome to the channel Microbial World. In this video, we will be studying about Streptococcus pneumoniae, its morphology, virulence factors, pathogenesis, laboratory diagnosis, treatment and prevention will be discussed. Streptococcus pneumoniae is a gram-positive lanceolate-shaped diplococcus. It is commonly referred to as pneumococci. Lanceolate shape means it refers to a shape that is narrow and tapering, resembling a spearhead. Okay? And these are normal inhabitants of the human upper respiratory tract. They are also bacterial agents in pneumonia and in otitis media in children. So pneumonia you have heard commonly. It is an inflammatory condition of the lungs affecting air sacs. Whereas otitis media is an inflammation or infection in the middle ear. It can cause ear pain and sometimes it also affects the hearing capability. They can also cause sinusitis, bronchitis, bacteremia, meningitis and other infections. Sinusitis means it is an inflammation of air-filled cavities around the nasal passages. Bronchitis is the inflammation of bronchial tubes which carry air to the lungs. And bacteremia is the presence of bacteria in the bloodstream. Meningitis is the inflammation of the membrane surrounding the brain and spinal cord. Okay? Morphology. So as we said before, they are gram-positive bacteria. They may lose gram-positive character in older cultures and appear gram-negative. They are typically small, slightly elongated cocci with one end broad or rounded and the other pointed, presenting a flame-shaped or lanceolate appearance. They occur in pairs, that is why they are called diplococci. They are capsulated, the capsule enclosing each pair. The capsule may be demonstrated as a clear halo in India ink preparations. They are non-motile and non-sporing. Virulence factors. Polysaccharide capsule is the major virulence factor of Streptococcus pneumoniae. Antibody to the polysaccharide capsule provides type-specific immunity. Peptidoglycan or teichoic acid are highly inflammatory in the central nervous system. Teichoic acid also helps in adherence. IgA protease helps in colonization. Pneumolysine O. It is a membrane-damaging toxin produced by Streptococcus pneumoniae that has cytotoxic and complement-activating properties. It damages the respiratory epithelium and it also inhibits leukocyte respiratory burst. Streptococcus pneumoniae has another antigen called somatic C carbohydrate antigen. It produces C-reactive protein in the hepatocytes, that is the liver cells. Its production is stimulated by bacterial infections, inflammation, malignancies or tissue destruction. Pathogenesis. Streptococcus pneumoniae colonize the human nasopharynx and may cause infection in the middle ear, paranasal sinuses and respiratory tract by direct spread. Pneumococcal bacteremia may also lead to distant infections in the heart, peritoneum or joints. So this peritoneum, it is a thin transparent membrane that lines the abdominal cavity. Okay? And Streptococcus pneumoniae, it is one of the most common bacteria causing pneumonia, both lobar and bronchopneumonia. So this lobar pneumonia, it is the inflammation of entire lobe of the lung. The infection first affects one lobe of the lung and then it covers the lung tissue and impairs the respiratory function. And in case of bronchopneumonia, the inflammation starts in the bronchi and then it spreads to the surrounding lung tissue. Okay? So in case of lobar pneumonia, bacteremia is common during the early stage of lobar pneumonia. And bronchopneumonia, it always occurs as a secondary infection. Damage to the respiratory epithelium and excessive bronchial secretions caused by the primary infection will facilitate the invasion of Streptococcus pneumoniae along the bronchial tree. And this bronchopneumonia also occurs as a terminal infection in aged patients. Okay, and they also cause acute tracheobronchitis and empyema. So this tracheobronchitis, it is the inflammation of both the trachea, that is the windpipe, and the bronchial tubes. Okay, and empyema, it is the accumulation of pus in the pleural space, that is the area between the membrane layers covering the lungs and the chest cavity. Okay, and toxemia occurs due to the diffusion of capsular polysaccharide into the blood and tissues. Streptococcus pneumoniae, it is also commonly associated with acute exacerbation in chronic bronchitis. The respiratory secretions in chronic bronchitis will further aid the pneumococcal invasions. And another bacterium that is commonly associated with this condition is Haemophilus influenzae. Meningitis is another serious disease of pneumococcal infections. And this meningitis, it also occurs as a secondary infection to other pneumococcal infections like pneumonia, otitis media, sinusitis or conjunctivitis. And they also produce suppurative lesions in other parts of the body. And it is also responsible for ocular infections like keratitis and dacryocystitis. So this keratitis, it is the inflammation of cornea, and dacryocystitis, it is the inflammation of lacrimal sac. This lacrimal sac, it is a part of the tear drainage system in the eye. Okay. Laboratory diagnosis. Specimen: sputum, blood, endotracheal aspirate, bronchoalveolar lavage, cerebral spinal fluid, pleural fluid, joint fluid, abscess fluid, bones and other biopsy material are taken as specimen. Microscopy. Gram staining of sputum shows lanceolate shaped gram-positive cocci in pairs. Fresh emulsified sputum mixed with antiserum causes capsule swelling for the identification of pneumococci. It is called Quillen reaction. Culture. Sputum or blood is plated on blood agar and incubated at 37 degrees Celsius in the presence of 5 to 10% carbon dioxide. Grey colonies with alpha hemolysis are observed after overnight incubation. Identification of bacteria. Optokin sensitivity test. Streptococcus pneumoniae is identified by its sensitivity to optokin, that is ethyl hydrocuprin dihydrochloride. The isolate is streaked onto a blood agar plate and a disc saturated with optokin is placed in the middle of the inoculum. A zone of inhibited bacterial growth is seen around the disc after overnight incubation. Bile solubility test. It detects an autolytic enzyme amidase present in pneumococci. This amidase enzyme breaks the bond between alanine and muramic acid of the peptidoglycan of pneumococcal cell wall. After exposure to bile, autolysins are activated and isolates of Streptococcus pneumoniae are lysed rapidly. Thus, the organism can be identified by placing a drop of bile on an isolated colony. Inulin fermentation test. It ferments inulin and hence differentiate it from other streptococci. Animal inoculation. By intraperitoneal inoculation in mice, pneumococci can be demonstrated in the peritoneal exudate and heart blood of the mice, which die 1 to 3 days after inoculation. Antigen detection. Capsular polysaccharide can be demonstrated in blood, urine and cerebral spinal fluid by counter-immunoelectrophoresis. An immunochromatography based test is available for the detection of polysaccharide antigen in urine. C-reactive protein is used as a prognostic marker in acute cases of acute pneumococcal pneumonia, acute rheumatic fever and other infectious diseases. Antibody detection. Indirect hemagglutination test, indirect fluorescent antibody test, and ELISA, enzyme-linked immunosorbent assay, are used to demonstrate specific pneumococcal antibodies in invasive pneumococcal diseases. Nucleic acid-based test. Nucleic acid probes and PCR assays are used for the identification of Streptococcus pneumoniae isolates in culture. Treatment. Most pneumococci are susceptible to penicillin. Other antibiotics such as macrolides or selected fluoroquinolones are available for patients who are allergic to penicillin. Nowadays, penicillin-resistant pneumococci are being documented. So, most resistant strains remain sensitive to third-generation cephalosporins such as cefotaxime or ceftriaxone. Ceftriaxone can be used for meningitis. Vancomycin is used if the pneumococcus is resistant to ceftriaxone. Amoxicillin is the drug of choice for treatment of otitis media, sinusitis, and pneumonia. Prevention. Pneumococcal vaccines. 23-valent pneumococcal polysaccharide vaccine, PPSV23, and polyvalent pneumococcal conjugate vaccine, PCV13, play an important role in the prevention of pneumococcal diseases. Personal hygiene is another important factor. So this is all about Streptococcus pneumoniae. Thank you.